So what I'm trying to do in this hour is I'm going to try to wrap up 2 or 3 unrelated things, mostly because these are things that come up on STEP exams and they don't necessarily fit easily into other things. So I'm going to start off by talking about the aldosterone paradox, and then we'll talk about tubular flow and how it affects potassium handling, and then we'll talk about how the hormones are controlling potassium and sodium differentially. Then this will be a prelude tomorrow. We'll start talking about the concept of free water, because I know that they've asked you to do free water calculations. And then finally, we'll look at some single nephron concepts, because again, they've come up on STEP exams. So let us begin. You know, since I'm having a kind of a scruffy Monday, we can probably skip that case. What it was a case that was trying to demonstrate that there can be conflicts in the stimulation of aldosterone. So remember, I had said aldosterone does 2 things. It retains sodium and it wastes potassium. And needless to say, it's turned on by low ECF or low pressure via the angiotensin system but it can also be turned on by elevated potassium, okay? And all I'm trying to do is, when I take you through that case, is I'm going to ask the question, is it possible you can have a low pressure or low ECF and low potassium as well? And if that is true, how does the aldosterone system cope with conflicting regulation? So in other words, if we have, and this is, I mean, never mind this case, it is quite frequent. You know, person's on a diuretic regimen and he gets volume depleted, but he also gets potassium depleted. I mean, that would be true of a lot of diuretics, right? What happens to aldosterone? You've got something that wants to stimulate it here and something that wants to turn it off here. Well, as you probably guessed, the low volume trumps the potassium situation, okay? But, so in other words, the low ECF and low potassium will actually stimulate, so this will tend to be overridden. The low potassium will tend to be overridden. However, what is the consequence of that override? Well, presumably the aldosterone will now retain sodium, which is a good thing, but oh crap. We got low potassium and it wants to kick more potassium out. So we actually may have got ourselves into a whole bunch of trouble here. And so we need to look at mechanisms by which we can at least temper this conflicting situation. Okay, so let's flip through the page and go to the bottom of page 2. And that's basically the aldosterone paradox. And there's two things that I want to do. One is talk about tubular flow, and then talk about the hormonal approach. Let's talk about tubular flow first. So this is a principal cell. So we turn on this cell, which seems appropriate because we want to retain sodium. So how do we not waste potassium? Let me rub this off. This is getting in the way. Well, the first thing is, remember I said that we had volume depletion. So volume depletion, the GFR is probably low, yes? So with a low GFR, the tubular fluid that arrives at the principal cell, tubular fluid is low. In other words, there's very little tubular fluid flowing past here. What this effectively does is that because there is less fluid here, as potassium is secreted, there is less fluid to run it away, and therefore the concentration gradient builds up and restrains the movement of potassium out to here. So in other words, normally you have a very high potassium inside the cell, relatively low outside, and so this moves fairly quickly. And if it gets washed out, you can maintain that gradient. But if you've got low tubular flow rate and it's just easing through here, as soon as you secrete potassium, it's going to raise the concentration gradient and it's going to restrain this passive movement. Okay? So what I'm saying is that the low tubular flow rate in volume depletion actually prevents extraordinary losses of potassium. Also remember that on the blood side, remember we said that there was low potassium? So the total gradient from here to here is also pretty low. So that will further hinder the secretion or movement of potassium from here to here. Okay? So I'm not telling you that it turns off potassium secretion completely, but it does at least restrain the potassium losses. Okay? So low tubular flow rate restrains potassium losses. And remember back when we talked about renal failure, one of the problems was getting rid of potassium. Here we actually have potassium depletion, and so it's kind of a good thing. But if we had renal failure and a low tubular flow rate, and we're eating lots of potassium, we want to get rid of it, this would actually be a hindrance, wouldn't it? And indeed, in renal failure, one of the issues is that you don't have enough GFR and tubular flow to wash out the potassium. And so potassium can build up. And I don't remember exactly, but we had that, I think it was a case of polycystic kidney disease, and I'm pretty sure the potassium was pretty darn high. Yes? Okay. So, any questions on that? That's a concept that you've got to be able to get your hands around for step exams. Okay. Let's move on to the aldosterone paradox. And I think I'm actually going to show a slide even though I told you before I'll never show another slide, I lied. So we now have this diagram actually came out of Nature. Let me turn this back on. Okay. This actually came out of Nature about 8 or 9 years ago, and I redrew this in the handout. And what I have here is, I have two situations. I have basically the whole renin-angiotensin-aldosterone system in hypovolemia, and I have the same thing here set up for hyperkalemia. So two different situations. And I want to convince you that these two situations actually behave differently. What I've also done is I've actually put most of the significant sodium transport mechanisms in, starting at the proximal tubule, the sodium hydrogen exchange, the NCC, which is in the distal tubule, the ENaC and the ROMK, which is in the principal cells, and then stuff going on in the intercalated cells. Okay, so in hypovolemia, or low blood pressure, whatever, you get stimulation of renin. That stimulates angiotensin II, and it turns on a lot of sodium reabsorption. First of all, it has a direct effect on the proximal tubule, and that's fairly pronounced. So retaining sodium here. It also, this is something we haven't mentioned before, there's also good evidence that it turns on the NCC. And it also turns on aldosterone, and the aldosterone turns on ENaC in the principal cell. But at the same time, rather than wasting potassium, the angiotensin II actually turns off the ROMK channel a little, okay? So everything is geared up to primarily replenish the hypovolemia and at the same time restrain the system from excessive losses of potassium, okay? In the other situation with hyperkalemia, whoops. Same picture. Hyperkalemia actually turns off the renin-angiotensin system. Okay, there's no hypervolemia. There's no hypovolemia here. Hyperkalemia actually turns this off, but it does turn on aldosterone. And as you might imagine, hyperkalemia turns on aldosterone, and it turns on this exchange between sodium and potassium. And so potassium secretion happens. Interestingly, this pump back here actually gets turned off by high potassium, and that makes sense, and I'll tell you why in a little bit. And since we have no renin-angiotensin system, we're not reabsorbing sodium as much in the proximal tubule. So how does this piece together? Well, if you've turned off sodium reabsorption here, and you've restrained it here, then by the time you get to the principal cell, you really need to reabsorb some sodium. And so now you're reabsorbing it with something that wants to exchange with potassium, and everything fits in. So you haven't excessively reabsorbed sodium because you've turned it off here and turned it on here. And as you turn it on here, you get rid of the potassium that you wanted to in the first place. Okay. The details of this are not terribly testable. Okay. But I think it's worth spending the 5 minutes that we did just so that you understand that we can actually handle these conflicting signals. Okay? Yes, ma'am. Question.
Yes. So during hypovolemia on the graph, you say that angiotensin 2 inhibit ROMK, but aldosterone stimulate ROMK. So, so which is that way?
Well, so the question is that here we turn on ROMK. Let's go back up. Okay. So you're basically just concerned about this conflict here. And obviously it's a, I'm sure that there's going to be some potassium loss, but it's restrained. Okay, so instead of being this massive potassium loss, it's restrained by the inhibition of angiotensin 2. Okay, and I can't. It's not like my taxes. I can't get the exact numbers for you. Okay, I'm not sure I can for my taxes either. But okay. So we good? All right. Okay, we're going. If there's no questions on that, we'll move on to free water. So I'll turn these things off. You get about 1 minute rest. So tomorrow we are going to talk about ADH, and ADH is all about the control of free water and osmotic pressure. But I know that the STEP exams kind of like you to be able to work with some of these things, some of these calculations, so we're going to go through them. The concept of free water balance, or free water clearance. So I've got 3 urine samples here collected on 3 separate days. On this day, 300 milliosmoles were lost in 2 liters of urine. On this day, 300 milliosmoles were lost in 1 liter of urine. And on a relatively dry day here, 300 milliosmoles were lost in half a liter of urine. So, based upon what we've done before, what's the osmolar clearance? It's the volume that those 300 milliosmoles occupied while they were in plasma. 1 liter! What's the osmolar clearance of this 2 liters? 300 milliosmoles, 1 liter. Uh-oh! 300 milliosmoles originally resided in 1 liter, so the osmolar clearance is 1 liter. There's an additional liter, but it's not the osmolar clearance. Okay, I bet you can get this one, and it's not really a candy one. 300 milliosmoles In half a litre. The clearance is 1 litre. Okay, good. So they're all 1 litre osmolar clearance. How much free water did I take out of plasma? None. Yes. Taken over. Yeah. You know, all the water here was basically associated with those 300 milliosmoles. And so there is no free water gained or lost. And I know this is easy, but, you know, bear with me. Let's do it anyway. The osmolar clearance is 1 litre. We lost 1 litre of free water, didn't we? Clearance of 1 litre, osmolar clearance of 1 litre. Therefore, we added negative half a litre of free water to the urine. Added negative, or we lost half a litre of free water, right? Okay, let me slow down then. So the osmolar clearance, that was associated with 1 litre of plasma, and we've only got half a litre of urine. And so the free water, I guess it's still in the plasma, right? Half a litre of free water is basically added back to plasma. So it's negative half a litre in the urine. Yes? Okay. So free water clearance equals urine volume minus osmolar clearance. Okay. So this is 2 litres minus 1 litre, which is what we came up before, right? Yes? Okay. Now understand that this truly is an osmolar clearance. This really isn't quite a clearance, but it's called free water clearance. Okay. And you guys are M1 students, and so you're supposed to be able to calculate this in step exams. So, do we actually ever do this clinically? There's a lot of stuff that you do in the first 2 years that probably doesn't directly relate to anything that you're going to do. So if you wanted to get an estimate of how much free water I was losing or gaining, are there any clinical tests? The most obvious one is urine-specific gravity, right? So specific gravity is basically just the density of the fluid. And pure water has a specific gravity of 1 because it's 1 gram per cc or per milliliter. And as you add solutes to it, the density increases, and so the specific gravity increases. So that would be a pretty good test, right? You just get a bob and you float it in the urine and see how much it floats and get basically an estimate of the salinity or the density of the urine. And that's it, that's specific gravity. You just put a bob in it and see how much it floats, and that's it. Can you figure out what the problem is with specific gravity of urine? Are there some diagnostic pitfalls? Right, it doesn't discriminate the type of solute. And that could be important because one molecule is one osmolar equivalent, right? But is a molecule of protein heavier than a molecule of sodium? Sure is. So in other words, if you have a lot of protein in the urine, it'll elevate the specific gravity immensely, and that really has no bearing on the osmolarity of the urine. It'll make it denser, but it's still only, a few protein particles. So, you would have to be cautious there. We can actually measure osmolarity of urine, and it's not an elaborate test. You just stick it into a machine, and it takes about 2 or 3 seconds, and it'll give you an exact value for osmolarity. And it's not affected by protein. Okay. There is also, did I write this down? Oh, I can go through this later. So let's go on to free water clearance, which I've already said that we don't use. So we're now on page 6, and I thought that I would just go through some numbers here that represent the extreme ranges for Curly, Larry, and Moe. So let's start with Curly. Curly made 3 liters of urine. Let's assume that his urine osmolarity, have I given you a value? Oh, 300, yes, 300 times 3, and let's always make plasma osmolarity 300. And if you calculate that, that's 3 minus 3 is 0. And we've already been through that here, and I think you probably already guessed that. Let us go and drink a lot of water. So Larry drank, golly, he drank 10 liters of water. And he made, probably more than 10 liters of water. So Larry has a urine volume of 10 minus the urine osmolarity. If he drank a lot of water, do you have any feel for it? It's probably going to be 50 to 100. Let's just say it's 100. 100 times 10 all over 300. And if you work that out, it comes to about 6.7 liters per day. So there's basically 6.7 liters of free water in that urine, which is presumably the amount that he drank, or at least close to the amount that he drank. On the other hand, we can actually conserve water and Moe, urine volume, have you discussed how much the kidneys can reduce their urine flow rate? You can't get much below 0.6 liters per day. Maybe 500 mils or half a liter, but somewhere around 0.6. In other words, if the kidneys don't make 0.6 mils of urine, they're probably not working properly. Okay, so urine osmolarity. Actually, this can go up to 1200, and we'll see this tomorrow. Oh, I've got a figure of 1400, okay. Probably 1200 in humans is about right. Times 0.6, all over 300. And if you work this out, I've already worked this out, it comes to minus 2.2 liters per day. So you're going to have to be able to know how to calculate those things. We tend not to like negative numbers, and sometimes that 2.2 is converted to a plus 2.2 tubular conservation of water. Okay, so TCH2O. It's just a conversion of the sign. Okay, and I only mention that because it's mentioned in some books. So having said all that, it's not really ever done because it's kind of a pain making those collections. I've just got at the bottom, you don't need to remember this formula, so I'm not going to write it up on the board. Okay. In clinically, you can actually calculate a free water deficit by looking at the plasma sodium. And that kind of makes sense. Remember, I'd said that sodium concentration, for the most part, is basically a product of water gain or loss, not so much sodium gain or loss. And so you can just plug in the plasma sodium into that formula and make an estimate of the total ECF volume, and you can actually come up with a figure that shows you. And I did a calculation. Is it on the next page? Okay. I don't think we need to do a calculation, but for example, if you have a sodium of say about 100 and say 160, that might represent a free water loss or deficit of 3 liters or so. You can actually do the calculation if you want. But we're not going to test you on that. It was basically just for your information, because one day you guys will all be doctors and you might actually do this one day. Okay, so one more thing. Anything on free water clearance? Okay. Single nephron concepts. So there's an old step question there. Let's see if we're any good at this. So I think Costanzo deals with this, tubular fluid to plasma ratios. So So plasma has an osmolarity of 300. Let's do sodium. Let's do inulin. So here the tubular fluid to plasma ratio for inulin, right here, right after it's filtered, what's it going to be? 1, yes. Okay. As you go along the proximal tubule, water gets reabsorbed, right? But the inulin does not. So the ratio has to start going up. Okay? And so the tubular fluid to plasma ratio of inulin is entirely dependent upon the amount of water that's been extracted from that tubular fluid. And that's kind of important because there's no reabsorption or secretion of inulin, and so the change in the tubular fluid to plasma ratio is entirely due to the amount of water that was taken out or put back into the tubule. Okay? So in this diagram, they're comparing substance X with tubular fluid to plasma ratio of inulin. If it behaves the same as inulin and it has the same ratios, it will mean that there is no secretion or reabsorption. If it deviates, it can only be due to secretion or reabsorption. Okay? So have we set the scene okay? So what are you thinking? In Bowman space, they're the same, right? 1. End of the proximal tubule. Oh, they deviate. So substance X has a greater concentration or relative concentration than inulin does. How can that be? It has to be secreted. And indeed, B is the right answer. It is secreted. So let's just go through this a little bit more. Well, wait a minute. It looks like, is something else going on? Well, not really. So at the end of the proximal tubule, it went up 4 times for substance X and 2 times for inulin. So we figured it's secreted. Okay, beyond that, X goes up 4 times at the end of the distal tubule, and it goes up 4 times for inulin too. So nothing else happened. And then just going further on, the ratio is the same from the end of the distal tubule to the bladder. Does that make sense? Okay, good. Okay, let me reproduce. I don't think this is in Costanzo. I think Costanzo has something similar. So what we're going to do here is we're going to plot tubular fluid to plasma ratios, TF to plasma ratios, and 1.0 here. And then we're going to go from one end of the nephron to the other. So this is prox, this is loop, distal, and collecting duct. So let's see if we can rationalize inulin first, which is green line, right? Green line. Obviously it starts here, and water's extracted in the proximal tubule, so it's going to go up quite a bit. What happens beyond the proximal tubule? Well, you go into the descending limb, right? And more water is extracted in the descending limb. Let me divide up this this loop of Henle. So you've got descending, ascending. And so what happens when you get to the descending limb? It should continue going up because you're extracting water. Okay. What happened now? I don't know whether it's clear here. In the ascending limb, it really shouldn't go down, should it? Shouldn't do that, yes? Because there's no water extracted. Then what happens in the distal tubule and the collecting tubule? Well, you continue to reabsorb more water, and so it continues to go up. Okay? So that's how the tubular fluid to plasma ratios of inulin work. And so if you wanted to, and you wanted to compare something to inulin, you just use this inulin pattern as a reference. The creatinine is in blue, and it behaves almost the same. Except remember, what is it, blue? Actually goes up just a little bit because some secreted, yes? But then beyond that, it just follows that. What shall we do next? Let's do PAH, which is a red line. And I don't need to really draw it in, but PAH is secreted a lot, and so it's going to do that, as it shows on the graph. And then once it gets to the end of the proximal tubule, it'll follow the same sort of pattern, albeit at an elevated level. Sodium, blue. Well, let's do this in red. Do you understand why sodium does that? It's isosmotic reabsorption, yes. Good. Okay. And then in the descending limb, you get water reabsorbed, but no sodium. So, gosh, it should go up. Does it go up? Where is sodium? Yes, it does. Okay. Yes? Makes sense? Now, what happens in the ascending loop? You get sodium reabsorption, but no water reabsorption. And that's why sodium goes down here. Okay? And then again, sodium and water, the sodium reabsorption exceeds the water, especially in the early distal. Actually, so it actually comes back up to there. Okay. Everybody follow how tubular fluid to plasma ratios work. I know it's something, it's listed in Costanzo, and it sometimes comes up on tests, on step exams. Now you could argue, why on earth, I want to be a doctor, why on earth do I need to know this sort of special vocabulary? And the answer, I don't have the answer for you, but as you know, sometimes you just kind of suck it up and do what they make you do, okay? And I hope that doesn't sound too cynical. Some of what they ask you to do, I think, is appropriate. This, maybe not so sure, okay? So, any questions? Well, it was a bit sluggish today, wasn't it? Sorry. You guys started off, you were looking good, and was it me? Okay. All right, any questions? Go, we'll do ADH tomorrow. ADH is actually it's fairly straightforward.